pleasant day to everyone. I am glad that you have reached this far into the course Epidemiology and Research Methods 1. Again, this is Dr. Kat Lee from the Department of Preventive and Community Medicine. This lecture is just a summary of what we have learned from this semester. Let's So for our learning objectives for this session, at the end of this, the learners will be able to be familiar with the different research designs and identify the different parts of research proposal and research paper. Now we go to qualitative approach, where qualitative data are usually found in words and are hypothesis generating. Here are four of the most common qualitative research designs. First, we have phenomenology. Its purpose is to describe experiences as they are lived. So it aims to find the essence or structure of an experience by explaining how complex meanings are built out of simple units of inner experience. Say, for example, the essence of being a participant in a particular program, or the essence of understanding a particular topic or subject. Next, we have grounded theory, and it derives from collected data, a theory that is supposed to be grounded in the data. So its purpose is theory development, and it's used in discovering what problems exist in a social scene and how persons handle them. And it involves formulation, testing, and redevelopment of propositions until a theory is developed. Next, we go to ethnographic research. Its purpose is to describe a culture's characteristics. In case study research, its purpose is to describe in depth the experience of one person, family, group, or community, or institution, just as in the case study in quantitative research. So here are non-conventional quantitative studies. First, we have the systematic review and meta-analysis, the diagnostic accuracy studies, and the validation studies. More on this later as we go along the presentation. Here are examples of health outcomes research designs. So under pharmacoeconomics, we have cost-benefit analysis, cost-effectiveness analysis, and cost-utility analysis. So cost-benefit analysis analyze costs and benefits of an intervention in monetary units. In cost-effectiveness analysis, it examines both the costs and health outcomes of one or more interventions. So the outcome is a ratio of the alternative interventions. In cost utility analysis, this is just another form of cost effectiveness analysis. So under modeling methods, we have Bayesian or simulation research. So in Bayesian research, it empowers decision makers to discover what most likely works by putting new research findings in context of an existing evidence data or evidence base. So next we have patient-reported outcomes, and a form of this would be your pharmacovigilance reporting. Next we have the health technology assessment. So during the in the Philippines, during the preparation for UHC, the creation of the HTA is to identify and recommend the most cost-effective health technology. So it includes medical, social, ethical, and economic dimensions for informed decision making in health. Next, we have patient preference research, under which we have utility measure or the quality studies and DALI studies. So, for quality adjusted life years, this is a measure of how many years of life are lined in good health. And in the disability adjusted life year, this is a measure of health, of lost health life in years. In the outcomes research, we have the meta research or the prognostic research. So here is a general format of a research paper. So a research paper is written after the research project is completed, and it's focused to present findings and subsequent conclusions. Here are the parts of the research: title page, abstracts, which contains also the keywords, introduction, materials and methods, results, discussion, references, and appendix. At the last, we have the author's curriculum vitae. So, earlier, a research proposal would usually be written before the research project, and it is written usually to seek approval or funding. So, we also have the title page, the abstract, the introduction, and here we have the review of literature. So in a research paper, this is already conjugated in the introduction. So the review of related literature is the appraisal of the studies with similar population, intervention, or exposure and outcome as your research questions. So we also have the methods. The research proposal is more rigorous, and it proposes the time, the personnel and facilities and budget that it will probably need for the research proper. Of course, we have the citation of references and also the proponent's curriculum vitae. We have checklists for the different research designs. So let's start with case report and case series. So we have the CARE checklist. So CARE stands for Case Reports Guidelines, developed by an international group of experts to increase accuracy, transparency, and the usefulness of case reports. The CARE guidelines for case reports help authors reduce the risk of bias, increase transparency again, and provide early signals of what works for which patients under which circumstances. So, important here is the patient information. Of course, it is reporting a case of a certain patient. So, the patient-specific information is often de-identified. So next, we go to observational studies. So in observational studies, you start with your target population. Then from a bigger target population, you select a smaller sample population using your inclusion and exclusion criteria. Then this sample population are grouped either with or without the risk factor or without the disease or outcome. Case control studies usually begins with two groups. The case are those with the disease or the outcome of interest, and the controls are those without the disease or the outcome of interest. In this study, we used the GWA, either high or as the case, and low GWA as the controls. So the outcome is already present at the start of the study, and then comparison of past exposure of both groups are extracted and compared. So for this study, the exposure would be if they are coffee drinkers or non-coffee drinkers. So in cohort studies, this is also an observational study design which is used to determine association between exposure and disease. So it starts with a cohort of participants at present time with exposure, or a similar cohort comparison group without the exposure. So for this example, we have coffee drinkers, coffee being the exposure, and non-coffee drinkers, those without coffee. And they are followed over time for the outcomes. So the outcomes here is pass the boards or fail the boards among coffee drinkers and those non-coffee drinkers. So the next checklist we have is the STROBE checklist for observational studies. So STROBE would mean strengthening the reporting of observational studies in epidemiology, or STROBE. It is a guideline on reporting observational studies such as cross-sectional studies, case control, and cohort studies. It is also created to aid the authors in presenting their work validly or reliably. So we have the title and abstract, introduction, where we have the background and objectives. We have methods, the results, the discussion, where we have the key results, limitation, interpretations, and the generalizability in a bigger population. Continuing, we have the study design, the setting, the participants. So for cohort, we have the eligibility criteria and methods of follow-up. And for case control, we give what is the definition of being a case. So next, we have the variables, bias, sample size, statistical methods, so under which we explain how missing data is addressed. We explain how loss to follow-up is addressed, particularly for cohort studies, and explain how matching was done, particularly for case control studies. So we have also the results. So the participants, we have the flow of diagram until their dropout or their continuation into the study. We have the descriptive data, the demographic characteristics, the outcome data or the number of outcome events, and the main results. We identify confounders and extract the relative rates for clinical trials, and odds ratio for case control. So other analysis, we have subgroup analysis. For clinical trials, we have, so here are other experimental studies that I have mentioned earlier. So we have the therapeutic trial. It's a phase 2 trial. So this phase 2 trial I will touch on later. And the therapeutic trial is used to collecting information on the potential efficacy of a new treatment or drug. So field trial are real-life experiments which test directly whether proposed interventions actually work. They usually gather evidence for making policies or health policies. Of course, I have mentioned community trial earlier. And lastly, diagnostic trial. So it is a study dedicated to assessing diagnostic methods in disease detection. We shall discuss this further as we go on. So here are selected concepts in the design of trials. So of course we have the control group, and this is the arm in a trial that does not receive the intervention. So it could be a placebo, as you have learned early on, or over standard versus standard of care. Next, we have randomization, and it is a method that ensures that participants have equal chances of being selected to an experimental group. So this is the most reliable method of creating homogeneous treatment groups, which would in turn minimize potential biases. So we have the admissibility criteria, where you have your detailed inclusion and exclusion criteria. So remember that exclusion criteria is not the opposite of your inclusion criteria. So they are also participants that pass the inclusion criteria but have additional characteristics that could interfere with the success of the study. These additional characteristics are supposed to be included under the exclusion criteria. Next, we have outcome ascertainment. So this is the complete capture of the outcomes of interest, and this involves a wide range of events from which the outcomes of interest are further evaluated and classified. Next, we have the ethics. Of course, you know that already. The good clinical practice, or the GCP training. This is an international ethical and scientific quality standard for the conduct, performance, monitoring, documentation, analysis, and reporting of clinical trials. And of course, this GCP is there to protect the rights and confidentiality of the trial participants. So experimental research, as we have mentioned, there is manipulation of the experimental group versus control group to establish a cause and effect relationship between variables. So it often uses controlled environments and random assignments to ensure the validity and generalizability of the research. So like the cohort, it go forwards in time. So we go to the drug development process. So long before any drug development and manufacturing can be done, researchers must identify or discover a specific molecule. So there are 4 phases of drug development. We have the drug discovery, preclinical studies, then clinical development, and finally the market approval, where we have the FDA review and large-scale manufacturing. So in the discovery stage, it starts with research and development in a laboratory setting. So researchers, they usually identify target molecules such as genes or proteins that are believed to have potential to contribute to the development or aggravation of a disease. So usually 5 to 10,000 compounds are being tested. Next, we have the preclinical studies, where the lead compounds are tested both in vitro and in vivo. So experimental models that come as close as possible to resembling humans are used in vivo studies. So here we use animals. Once fully characterized, the most promising compounds become lead candidates. So the most important aspect of this part in preclinical research is the rigorous safety test with the purpose of ensuring that the candidate drug is not toxic before it can go through clinical studies in humans. So altogether, this discovery phase and the preclinical phase can take 3 to 6 years. So then after the completion of the preclinical tests, provided that the results positively answer the research hypothesis, the developers will apply for permission to proceed with clinical studies in humans. So under the clinical development, we have four phases. Phase 1, we have the safety. Phase 2, proof of concept. Phase 3, we have regulatory evidence. And phase 4 is post-marketing surveillance. So following regulatory approval and approval from ethics committee, the first clinical study, phase 1 study, which constitutes the first study in humans, is already initiated. So here, the candidate is generally tested on 20 to 80 healthy volunteers with the goal of determining whether the candidate behaves in the same way in the human body as the preclinical studies have indicated. So the safety profile of the substance is again the main focus, but this time it is done in humans. So in phase 1, you test what constitutes a safe dose, how drug is absorbed, and how long it is active in the body. So it is worth noting that for safety reasons, phase 1 clinical trials tend to exclude women of childbearing age because of this purpose. So it takes about up to 1 year to perform. Next, we have phase 2, proof of concept or effectivity studies or efficacy studies. So in the event of positive safety results from phase 1, the drug developers now can apply for permission to take the next clinical development step. So, which is the phase 2. In this phase, the candidate is most often evaluated in 100 to 300 patients diagnosed with the disease that the candidate is intended to treat. So when we say candidate, it is the lead drug. So here, efficacy joins safety as minimum and maximum dosages of the drugs are determined for use in the next phase of development. So phase 2 typically takes up to 2 years. Next, we have the phase 3, the regulatory evidence. So here, in the case of positive safety and efficacy from phase 2, the next step is phase 3 already. So this is the last step in the evaluation of a drug before requesting market approval from pharmaceutical regulators. The number of patients enrolled in this phase of the study is usually at least 1,000. This ensures that enough data is obtained to show that the drug is safe for humans and has intended clinical efficacy. So, in connection with phase 3 studies, researchers document and report any side effects experienced by patients. So this means that patients need to be exposed to the drug for longer periods of time in order to make sure those side effects are correctly assessed. So any side effects noted at this stage are those that are listed already in the package leaflet of the final product. So again, this takes on average of 1 to 4 years, so it takes longer. At this point, there is the drug registration process. So in the event we have good results in phase 3, we have an application for market approval already. So this can include hundreds of thousands of pages of documentation which summarizes all collected data from the discovery phase onwards and where the principal investigators argues for approval, usually with the FDA. If the regulatory authorities approve an application, the candidate drug or medicine, as is now called, is ready for market launch. So going back to the checklists, in clinical trials, we use the Consort checklist. Consort stands for Consolidating Standards of Reporting Trials. So it is a 25-item checklist that provides the author with a solid backbone around which to construct and present a randomized controlled trial. So it sets standards on the trial's design, analysis, and results. So we also have the title and abstract, introduction, methods, results, discussion, under which we have limitations, generalizability to a bigger population, and the interpretation of those results. Other information would include the registration of the product, the protocol, and the funding. So under the methods, we have the trial design, participants, the interventions, the outcomes, the sample size. Of course, this is important in randomized clinical trials, the randomization, wherein we have usually the sequence generation, which is a method of generating the allocation sequence. Example, the usage of computer generated random numbers, of course, among others. We have also the blinding and the statistical methods. So blinding of the outcome assessors, blinding of the participants, and the caregivers. In the results, we have the participants' flow. So we have the diagram or flow chart. We have the recruitment, where we have the inclusion and exclusion criteria, the baseline data, where we have the demographic and clinical characteristics if they are comparable. We have the outcomes and estimates, some subgroup analysis, and some harms that were noted during the study. So systematic review, they include randomized controlled trials with similar population, intervention, outcome as your study participants. So systematic review attempts to gather all available RCTs by using clearly defined systematic methods to obtain answer for a specific research question. A meta-analysis, on the other hand, is the statistical process of analyzing and combining results from several similar clinical trials which you selected in the systematic review. So, Review Manager is Cochrane's software for writing reviews and meta-analysis. So, it supports the development of evidence synthesis and can be used to generate forest plots, among others, meta-analysis, and risk of bias tables. You use this to make your systematic review and meta-analysis protocols, and this is all that you will need in doing a meta-analysis. So this is usually a web-based software. This is the process of meta-analysis, which we shall discuss in depth in research methods too. But it usually starts with the formulation of the review questions and the inclusion of the studies using the inclusion and exclusion criteria. And then we develop search strategies and locate those studies. We select the studies and extract the needed data from the studies. Next, we assess the study quality, and we usually input the extracted data in RevMan, where the RevMan analyzes and then interpret the results. From this interpretation of the results, we now write the whole meta-analysis extracted from RevMan and disseminate the findings of these different randomized controlled trials included in the meta-analysis. So it also has a checklist. So the Prisma checklist, this stands for Preferred Reporting Items for Systematic Reviews and Meta-Analysis. So it is a 27-item checklist addressing the introduction, methods, results, and discussion sessions of a systematic review report. This is already the standard format for reporting systematic review and meta-analysis. So we have the title, the abstract, introduction, methods, results, discussion, and finding. So under the methods, we have the protocol and the registration, eligibility criteria of the studies, information sources, the search strategy we use, the study selection process, the data collection process, the data items where all variables are sought, the risk of bias, this is very important in meta-analysis, and of course the methods used to assess the risk of bias is important. Uh, Summary measure, it is the effect size used across all studies and the synthesis of the results. So in the results, we have first the study selection, and it's the number of studies screened. So study selection is a flow diagram of numbers of studies screened until we have the final list of studies. Then we have the study characteristics of those already selected, the risk of bias using the funnel plot. We also report the results of individual studies, the synthesis of these results using the forest plot, and additional analysis. So if there's any sensitivity or subgroup analysis where there is trend observed among certain studies that were included. So here we have the forest plot. The forest plot is a tool summarizing information across different studies that were included in the review. At the far left are the different studies that were included in the review and the year they were published. So it gives a visual suggestion of the amount of heterogeneity and show the estimated common effect in one figure. So the diamond here represents the point estimate and confidence interval when you combine and average all the individual studies together. So the vertical line here is called the line of nullity. So anything that crosses the line of nullity does not depict a statistically significant results. So since they all cross the line of nullity here, none of the studies depict statistically significant results. So here at the bottom, you have the I2 statistic. It gives us the idea of the heterogeneity of the studies. So if it's more than 50%, it might mean the studies are inconsistent due to a reason other than chance. But this will be discussed further in research methods too. So next we have funnel plot. So this is an example of a funnel plot. So a funnel plot is used as a way to assess publication bias in meta-analysis. So the area here where it's relatively empty suggests that publication bias is evident. So validation studies compare the accuracy of a measure with a gold standard measure to identify and eliminate bias, and it usually is used to validate questionnaires as well. So this is a design framework of how to validate questionnaires. But we will discuss this again further in research methods too. So we start with the preparation, the forward translation of a questionnaire, the reconciliation, and the back translation. Next, we have the back translation review, the harmonization of the data, the cognitive briefing, the review of cognitive debriefing results, and finalization. And towards the end, we have the proofreading and the final report of the validation study. But anyway, this will be, as I said, this will be discussed further in research methods too. we go to diagnostic accuracy studies, and usually it evaluates the ability of one or more medical tests to correctly classify study participants as having a target condition. So how do we perform diagnostic accuracy studies? Again, we start with participants with the inclusion criteria. So the test whose accuracy is being tested is called the index test. So evaluating the ability of a medical test to correctly classify patients is typically done by comparing the distribution of the index test with those of the reference standard. So the reference standard is the best available method for establishing the presence or absence of the target condition. So again, an accuracy study can evaluate the accuracy of one or more index tests and can rely on one or more reference standards. So diagnostic accuracy can be overestimated if the test is evaluated in a group of patients already known to have the disease and a separate group of normal patients rather than in a relevant clinical population. This will be referred to as a case control study. So again, these are going to be discussed in detail on research design, but these are the things you need to look for. The sensitivity and specificity, you already know that. Positive and negative predicative values, the PPV or the NPV, the likelihood ratio, so we already discussed this earlier into the course. The area under the curve. So the area under the curve informs in a single numerical value about the overall diagnostic accuracy of an index test. We have the Udens index. It combines sensitivity and specificity in a single measure. So the formula for that is sensitivity plus specificity minus 1 and has a value of 0 between 0 to 1. It is a summary measure of the effectiveness of a diagnostic test or marker. We have diagnostic odds ratio. So the DOR of a test is the ratio of the odds of positivity in subjects with disease relative to the odds in participants without the disease, and depends significantly on sensitivity and specificity of a test. So again, you know this, it's the 2 by 2 table, and I think we have explained this, tackled this in depth during our preceptorial sessions. Diagnostic accuracy test also has a checklist. It's called the Standards for Reporting Diagnostic Accuracy Studies, or the START, and it is a 30-item checklist identified by an international expert group of methodologists. The QUADAS 2 is Quality Assessment of Diagnostic Accuracy Studies. It's a tool for systematic reviews of diagnostic accuracy studies and is used to evaluate the risk of bias and applicability of primary diagnostic accuracy studies. So here is the first parts of a START checklist. So title or abstract, introduction, methods. Under methods, we have the study design, participants, the test methods used, and analysis. Under the results, we have the participants versus the test results and the discussion of the results. For timelines, we have already discussed the different checklists. So all of these checklists will be found in the Equator network. So the Equator Network is a web-based one-stop shop for writing and publishing high-impact health research. It's like a library for health research reporting. So now we go to the research problem. Actually, in order to start a research, you should always have a research problem for a certain clinical scenario that you have seen in your practice. So we should have a research topic. It's usually a general area of study or issue, and a research problem. A research problem is a more specific question regarding your research topic. So here are important questions about research problem or the finer criteria. First, we ask if it's feasible. So we ask for the adequate, no, we think of the adequate number of subjects, adequate technical expertise, if it's affordable in time and money and manageable in scope. So we ask, can it be answered with scientific inquiry, or do I have the resources? Your research question should also be interesting to the investigator in particular. So you ask, does it really interest me? Is the study novel? Like, does it confirm or refute previous finding? Does it extend or previous finding or provides new findings? Is it ethical? Are there irresolvable ethical problems? It should be relevant to scientific knowledge, to clinical and health policy, and to future research direction. So, is it theoretically or practically important? So those are the questions that you have to answer in making your research questions. Now from the research question, we state the research problem. So we usually use the PCOM or the PIOM format to state a research question. So population, P stands for population, I stands for intervention, C stands for control or comparison, O stands for outcome of interest, and of course M stands for methods or research methods that was used. So sometimes it doesn't have a control, so it's called PIOM, population, intervention, outcome of interest, and methods. Or sometimes there is no intervention but an exposure. So it's called PEOM, population, exposure, control, outcome of interest, and methods. So format to stating a research question for PCOM and PIOM. So for PCOM, among the population, is the intervention better than the control in producing the outcome using this research method? So this should be the format. And in PIOM, among population, does the exposure lead to the outcome using method? So we have examples for this. Among newborns, is being delivered in CMONC facilities associated with lower IMR or infant mortality rate than being delivered in non-CMONC facilities in a case-controlled study. So newborn is the population, delivered in CMONC facilities is the intervention, delivered is in non-CMONC facilities is the comparator, and lower infant mortality rate is the outcome. Of course, case-controlled study is the methodology that was. So we have another sample for PIOM. Among patients undergoing repeated peritoneal dialysis, does the use of food supplements increase recovery rates in a longitudinal cohort study? So patients undergoing repeated peritoneal dialysis is our population. Food supplements is our exposure. Increased recovery rates is the outcome, and longitudinal cohort study is the design that was used. So I guess this is it. So I would like to part with a quote: "The real voyage of discovery lies not in seeking new landscapes, but in seeing with new eyes." So that's it for research review and consolidation. You may contact me for any clarifications or questions using my institutional email or via Canvas. Thanks so much.